Okay. Yeah, thank you, Aaron. And thanks so much for the invitation to speak at the OTS webinar. It's my first one, and it's very exciting to be here. So it's nice to tune in virtually with everyone here. Yeah, so like Aaron said, if you want to reach out to me, if you have any follow-up questions or anything after the webinar, my email's here. And just a little bit about myself, in case you don't know me, I'm at Ontario Tech, which is in Oshawa, Ontario, Canada. And I've been here since about 2008. So I'm starting to become kind of one of the old guys in nucleic acid chemistry, but hopefully not too old. So, so what I want to tell you about today is some of our work that we've been doing for a number of years at controlling gene silencing with light. And we have some new in vivo data that I'll be presenting sort of near the end of the talk, in which we can sort of photo-optically control the gene silencing in these Japanese Medaka fish embryos. So I'll get to that. And on the left here is, I guess, sort of foreshadowing what I'll be talking about, but we're using this compound called azobenzene, which has a really nice ability to isomerize between cis and transform. And we go through three different derivations in order to redshift it and use sort of different colored lights to be able to control that selectivity. But before that, I sort of want to tell you a little story about my group a little bit and how we led and how our designs eventually led to this photoswitchable group. Okay, so our lab focuses primarily on RNA interference. And certainly this really doesn't need much introduction to this audience. So as you know, double-stranded RNAs are cleaved through dicer or to generate short interfering RNA fragments, or you can exogenously administer things like short interfering RNAs because these are substrates for the RNA-induced silencing complex, also known as RISC. And what happens is that once this binds to RISC, the passenger strand is removed, typically through cleavage at the central region by Argonaute 2. And then we have a guide strand that is then able to bind through a canonical base pairing to the messenger RNA that cleaves the messenger RNA. And then we get no translation of the protein and hence gene silencing. So this is, this was sort of first sort of reported by Fire and Mello in 1998, and it's been really the thrust of a lot of pharmaceutical successes in recent years. So since 2018, there have been 6 US FDA approved siRNA therapeutics, so about one per year. I don't have time to go through the details of all of them, but suffice to say they target typically rare genetic disorders, sort of mutations, and they target the primarily the liver. So they're very organ specific. So despite the fact that we have actually 6 US FDA approved therapeutics, there's still mounting challenges with RNA-based tools and therapeutics. Probably one of the biggest challenges is, of course, stability. Here's the structure of RNA. And, as a chemist, we look at RNA and it's really a lousy looking structure. You can see the presence of this 2' hydroxyl, so the presence of nucleases. This is really accelerated to, get this nucleophile to attack this phosphate. So the RNA is rapidly degraded in cellular systems. Another big issue is cellular uptake. How does this negatively charged polyanion get across the cell membrane? So fortunately, there have been some really amazing discoveries, over the last decade and more with respect to delivering oligos to tissues such as the liver through nanoparticles, or more recently through GalNAc. And so that's been instrumental. However, there's still challenges with respect to understanding how the release of these oligos gets sort of released from the endosome once it's taken up in the cells. And certainly there are other organs as well that would benefit from things like siRNA treatment. Biodistribution, immune activations are also dominant issues in RNA-based tools and therapeutics. Issue that is also quite important are off-target effects. So siRNAs, once that passenger strand is released, you're left with the guide strand, and the seed region has the potential to, to pair with other similar sequence targets. So this could silence similar sequences, resulting in off-target effects. Or you could have the actual passenger strand, which should have been a passenger strand, actually being taken up as the guide strand, and then this can also induce off-target effects. So you can have these kind of like micro RNA-like off-target effects, and this can cause some serious issues as well. So specificity, cell membrane permeability, et cetera, are the hallmark of these issues with RNA-based tool therapeutics. And I just want to this was a nice paper that was published earlier this year, kind of highlighting the different chemistries used for RNA therapeutics, and just want to kind of briefly highlight some of the common players. So, siRNAs are heavily modified in order to overcome some of those issues I talked about, and sort of main players include things like 2' methyls, 2' fluoros. These help stabilize the RNA into the C3' endo conformation. There's certainly areas that could be used, like glyconucleic acids, to help with balancing thermal stability. There's a lot of intricacies with respect to, how active is that strand. So vinyl phosphates have been used to be placed on the 5' end of antisense strands to avoid kinases from removing them, so to increase potencies. Inverted abasic bases are used to help with strand selection. And then we have some of these carriers like GalNAc or other lipophilic ligands. But what I just sort of want to highlight here is that, we're playing with I would say, a relatively small sort of chemical playground of chemical modifications within the RNA field. So I think in order to overcome a lot of the current challenges, we'll need to expand the current repertoire of chemical modifications. So just before I talk about some of our work with azobenzene, I just want to kind of walk with you very briefly through some of the work that we've done over the years. We've taken look at things like triazoles and amide bonds to replace some of the phosphate groups, because in our group, we want to assess biocompatibility of chemically modified RNAs. More recently, we've been really interested in receptor-mediated endo uptake or endocytosis of oligonucleotides. And a few years ago, we published a paper looking at folic acid conjugated to RNAs, and we saw very good, efficient knockdown in transfection-free media. And, if anyone's interested in sort of learning more or looking at opportunities in my lab with respect to this project, please feel free to contact me. I don't have time to talk about that today. We have a collaboration in Japan with Professor Okauchi, who's really developed a nice method to develop asymmetrical triphosphate esters, or triester phosphate esters. And essentially what we've done is we've designed these molecules here with high hydrophobicity, and we've observed that many of these have a great cell membrane permeability because of their hydrophobicity. So these could address some of the issues with delivery. A recent graduate from my lab, Ifrit Georgies, has designed these Corol molecules, which are really interesting. They're photosensitizers, and they release reactive oxygen species. So some of our work has also involved using these types of compounds in combination therapy, that is silencing, and then using red light to sort of further treat cancer cells. But a lot of these projects really stem from an important discovery that we made about 12 years ago, in which we replaced nucleobases with these very simple spacer linkages. And I'll tell you a little bit more about what we did with that to get to our azobenzene scaffold. But I want to bring you back to this slide, because I did tell you about these 5 issues. However, there's some other issue that is really, I think, the focus of this talk today, and that is being able to control the activity of an siRNA once it's inside the cell. So, there's been great sort of discoveries and anecdotes. We know that siRNAs, once they enter the liver through GalNAc, for example. They go through endocytosis, and then that siRNA can last for weeks or even months, right? So this is really good, meaning that we don't need to get injections every week, but we could actually get injections every few months. But however, this could also come at a cost in that you do then have this active RNA that's slowly being leaked from the endosome, but it could also be susceptible to maybe toxicity or off-target effects. And so there might be a need to be able to control the activity of an siRNA once it's already in the cell to perhaps inactivate it or subdue it a little bit. And I just want to highlight an important study from Alnylam in 2018, in which they designed an siRNA that targets TTR protein in mice. And they observed that these mice were exhibiting hepatotoxicity, so toxicity of the liver, unfortunately, due to some of the toxic or off-target effects of the siRNA. So in order to reverse that, what they'd done was they designed oligonucleotide with a GalNAc, so it can get to the liver, that competes with the guide strand, and particularly the seed region of that guide strand attached to RISC. So this would presumably compete for that region, therefore disabling this ability on top to therefore bind to the mRNA strand. So what you can see here in the graph is that when they add the TTR siRNA, you get rapid reduction of TTR protein levels, and you can see that it remains sort of dormant or very low expression for, in this case, 2 weeks. So at day 7, they add Reversir, and you can see rapid sort of reversal of that protein, thus highlighting the need and the utility to be able to reverse the siRNA activity. And this was certainly an important discovery made by Alnylam and able to turn off the function of an siRNA. So back to sort of these spacer linkages that I want to just highlight to you about. Now, these spacer linkages, we made a discovery back, like I said, 12 years ago, and we hypothesized that if we put these spacer linkages within the central region of the passenger strand, that we could thermally destabilize them, and we could get sort of dissociative stand removal, but retain good RNA activity. And that's indeed what we saw when we placed either these linkages replacing 2 nucleotides, or this even longer linkage replacing 3 nucleotides. So to us, this was really interesting because this would potentially allow us to expand the scope of chemical architecture that could be used surrounding these linkers. So, I don't have time to go through all the different derivatives that we've made, but certainly, these different linkages worked. And then we looked at conformationally constraining groups like biphenyls, and those worked as well. So it really didn't seem to matter what we put at the central region within the scope. So we hypothesized that by placing azobenzene, maybe we could have a way to retain activity, but azobenzene being a photoswitchable molecule, maybe we could actually control the activity of the siRNA. So azobenzene, as I mentioned, just very briefly at the title page, is an interesting molecule. It isomerizes between trans and cis through UV light, and it's reversible, meaning that visible light or heat can go back. So it's an entirely reversible process, and it's been used widely in a variety of biological applications. So, it's been used a lot in peptide chemistry. For example, Woolley et al. A couple decades ago had this transform of this sort of long peptide, and then they could sort of force it into this alpha helix by making it go into a cis form. So we hypothesized that if we had our azobenzene in the trans form, then we could irradiate it with UV light. We could distort it, kink it in some fashion, rendering it potentially inactive. And then visible light could potentially reactivate that. So this was sort of our potential working model for this. And we're certainly not the first people to think of using like photochemical oligonucleotides or optical use of oligonucleotides. I'll just highlight some really seminal work by some researchers. For example, work by like Friedman co-workers. This is really clever, in which they put these nitrophenyl groups attached as protecting groups to these bases. So within the central region, this created a bulge within RISC. So this was inactive. However, with UV light, this these were removed, and then this initiated RNA interference. Sort of a more recent example, or sort of over the years, Mayer and Mokir moved away from UV light, and they used this anthracenyl group attached to the 5' end, and then through red light and through singlet oxygen species electron transfer, they could remove this anthracenyl group, resulting in sort of an uncaged siRNA that could then be active. So this was certainly interesting as well with an siRNA, going from something that's inactive to active with light or UV light. And this is certainly not restricted only to siRNAs. In this case here, we have a bicyclic morpholino, and this was recently published by Dieters and coworkers, in which they had this. It was held together through like a photolabile linker. So when they injected this in zebrafish embryo, UV light was able to linearize it, bind to the messenger RNA, and get gene silencing. So these are certainly very innovative techniques, but I would like to highlight that some of the limitations of these is that they are not reversible, meaning you can't go from an active complex to something that's inactive, and you can't go back and forth. So they were irreversible in their manner. So with our construct in place with azobenzene, we wanted to see whether we could at least mimic something similar to what I'd shown on the previous slide. So what we'd done was we synthesized active siRNA with that azobenzene, irradiated it, and then we transfected it. And then what we wanted to do is add visible light and see if we could sort of induce activity, a few hours after it had been transfected, to see whether we could sort of recapitulate some of those results from others. And what we've seen, and what we have here is a heat map, and I'll go over it with you. What we have is we really weren't sure exactly where to place azobenzene, so we sort of did like a short little kind of walk of this azobenzene scaffold across the central region of the scent strand. And then, so we had 4 different siRNAs, and then we looked at different concentrations. And then, so I'll bring your attention to this 8-hour assay here, where we had the UV inactivation, and we're looking at luciferase activity, or reporter gene. And rather than looking at bar graphs, I have a heat map. So sort of the kind of the more, or is kind of the lighter the blue, the higher the luciferase activity, thus highlighting that the siRNA is not working. And then the kind of the brighter the yellow, the lower luciferase activity, meaning that gene silencing is occurring. So what you can see from this collection here is that, the siRNA is really not working well because we see a lot of black and a lot of blue. Now, partway through this experiment, we add visible light to the cells that have been transfected with this azobenzene-containing siRNA, and then you can start to see some restoration of yellow, indicating that we're starting to induce some activity. So that's indicating that something's happening, which is very promising. And then over here is our what we call our dark control, meaning that this siRNA had never been inactivated before. So, these azobenzene we had shown, I don't have time to show the data, but they, on their transform and their native form, are a suitable substrate. So this is highlighting that these actually are working, and that this is very likely not an artifact. We could do this for up to 24 hours. However, one thing about this azobenzene group, and here's sort of our model, is that the cis form here will relax back with a half-life of around 4 hours. So in order to keep this inactive for 24 hours, we had to add short bursts of UV light every 4 hours. But you can see that it does work, but it's maybe not so practical. And then at any time point, a few hours later, we have visible light, and we can start to see restoration of RNA activity. And again, our control dark, showing robust gene silencing. And then here's our wild type control, showing that it's not affected by either of those conditions. So this was really promising. And then we want to now ask the question, well, could we go a step further, and could this process be reversible? So we asked the question, we could take in as active RNA that had the azobenzene, inactivate it, so kind of do the reverse of what I've done in that previous experiment, but then reactivate it with visible light. And then not only that, could we kind of go back again? Could we then re-inactivate it and then reactivate? So a lot of kind of shuffling back and forth. So basically 2-time cycling was what we were trying to achieve here. And so we did a 24-hour experiment. And, so if we look at our UV experiment, so, we just keep it inactive, short bursts of UV light every 4 hours, we can keep it inactive. This UV, visible UV, so we inactivate UV light, few hours later, activate it with visible light, and then a few hours later, re-inactivate it. So during this span, there'll be some activity associated because of the visible light, but certainly we might not expect it to be super strong. So we can see at lower concentrations, we really don't get good activity, but we do see some activity at higher concentrations. This next experiment is sort of the full cycle where we go from UV visible, UV visible. So we go through 2 cycles of recycling, and we would expect to see some level of ginseng because we've gone through 2 bursts of visible light, even though we've inactivated it, but it might not be sort of as strong as we might expect compared to dark. But we were very pleasantly surprised, or not surprised, but happy to see that we get good gene silencing and good dose-dependent gene silencing. And of course, when we compare that to dark, it's not as strong as dark, which is as expected because the dark never went through rounds of inactivation. And I just want to highlight that we can control the activation, but we could also potentially control the level of potency of the siRNA. So this also gives us sort of a spatial control within that siRNA duplex. Now, with this in hand, we did want to start moving away from UV light, however, because UV light, as you know, is really not ideal for biological systems because, it can cause TT dimer formation and it's really not practical. So we wanted to move away from, so once we established this proof of concept, we wanted to move away. And synthesis done by Dirk Schroner actually in 2016, they published a really nice paper in Chemistry, European Journal, in which they had done a post-synthesis functionalization with tetrachlorine groups. And so we kind of borrowed some of that chemistry to make it amenable for our synthesis, and we were able to synthesize, of course, our diol azobenzene, and then we used a tetrachlorinated group, N-chlorosuccinimide and platinum acetate to install these 4 chlorine groups, saponify the acetates groups off, and then we use this DMT group as a protecting group, and then phophoramidite, which is our building block for RNA synthesis. So in general, the synthesis was, it worked well. And it took some time to optimize it, but we were able to generate now tetrachlorinated groups, azobenzene groups within the RNA. So we did something very similar to what we had done with the initial azobenzene, in which we placed these tetrachlorine groups across that central region. And a little bit about this tetrachlorinated azobenzene group. So the advantage is that we can use red light to isomerize it to cis, as I'm showing you here, then we can use purple light to go back. So that is an advantage because we've moved away from red light. So let's look at some of the data. So if we take out siRNA1, and if we look at its activity with red light, you can see that we're not getting good activity. So that's great. Now, if we add thermal, so that is if we remove the red light and just let heat isomerize this back to trans, or if we keep them in the dark so they've never been exposed to red light, so they're transformed, you can see that we actually get good gene silencing. So we're looking at luciferase activity on the y-axis here. And we can do this for 24 hours as well, in which we can keep it inactive for 24 hours, and then we can get activity with thermal heat, or the dark experiments, you can see downregulation. Probably the main issue with this, however, was that the half-life for this tetrachlorinated group, going back to the sort of native transform, is actually really quite short, on the matter of really like minutes. And so this means that if you don't, what this meant was that in our experiments, we had to keep the cells under red light for the duration of the experiment. So red light's certainly not like toxic, and it's, maybe it's fun to be in a little red light chamber or something, but it's certainly maybe not very practical, right? So that was one of the issues with this. But you can see that it really didn't matter where we put this modification across the central region, we saw the same pattern, meaning that we could keep it inactive in all these different 3 RNAs, and then we could restore activity through thermal by removing the red light. And then our wild type control on the bottom here showing no effect with the red light. So this was certainly, like I said, a nice advancement, but not very practical if you're thinking of cell experiment or even potentially in a patient, who wants to be exposed to red light continuously if you want to control the activation. So so we kind of were, azobenzene was, we're kind of learning more about it. And we learned that there are these interesting compounds called tetrafluorinated azobenzenes. And this is a paper published by Feringa. It's nothing to do with siRNAs, but they were able to control the photoresponsive antibacterial agent by addition of these 3 different azobenzenes. And they noticed that, well, it was known that this tetrafluorinated group had a very long half-life and could be irradiated to give the cis form with green light. Okay, so that was very interesting. Some other reports by Feringa around a few years later showed that sort of this adenine derivative here, if they appended this tetrafluorinated group, irradiated to the cis form here, they could actually potentially control like the period and phase of circadian rhythms in cells in a reversible manner because they're inhibitors of casein kinase 1. And these could potentially, what they report, could be potentially used for things like combating jet lag if you go for a long flight or something. So these are very useful in that they were reversible, but they could be very long lasting as well. Some other uses of this group include, you have this scaffold here with this trans group, and it has this very defined gel-like shape. And then when you irradiate with green light, it kinks into this form and releases into now liquid. So you can see sort of differences in morphology from solid to liquid. And they had this molecule here, which was sort of hydrogen bonded in sort of this matrix here, and then through green light, this would collapse, and then you could get release of this molecule. So you could use it potentially as sort of a drug release form. But a lot of work done with tetrafluorinated azobenzene, but we hadn't seen its use within siRNAs, and we were very intrigued by its ability to be used for green light and long half-life. So I had a very talented visiting PhD student from QTech in Japan. His name was Kota Tsubaki, and at the time he was we were facing this problem, and he learned and helped our group develop a synthesis to make a tetrafluorinated group that was amenable for RNA synthesis. So I'll briefly go through this. Commercially available difluoroaniline, we can brominate this with NBS, do a cyanide sort of substitution, and then we can oxidize to carboxylic acid. And then what we've done here is we take this TBS protected linker, standard amide bond coupling conditions to form this group here. And then we did this diazo coupling with DBU and NCS at -78 using some recent conditions that were reported in Org by Antoine Lin for this symmetrical tetrafluorinated azobenzene. And really the rest is just protecting groove removal and manipulation. So TBS groups come off with acid, the DMT group, and then phosphoramidite to generate this phosphoramidite that we can now incorporate within the azobenzene or within the siRNA. So at this stage here, what we've done is our model is still the same. We have this tetrafluorinated group, add green light, inactive, and then blue light could potentially reactivate it. And in this case, we designed an siRNA with this azobenzene in the middle, but this time on the antisense strand across from the central region of the sense strand. So it's within this area here. So let's look at some of the in vitro data that we got for this. And so what we observed, okay, so 48-hour experiments. So what we've done, just want to say my former student and sort of current postdoc, Matt Hamill, spearheaded this project. And basically what he does, he transfected the cells and then added green light, and then basically kept the cells in the dark for 48 hours. So we wanted to avoid that continuous exposure of light because that's not very practical. And you can see here that the siRNA remains inactive after 48 hours after a single exposure of green light from the beginning onset. Now, this next experiment, green, blue, initially he added green light, and then a few hours later, he added a short burst of blue light to hopefully, make this molecule go back to the transform and restore activity. And that's exactly what we see. We see gene silencing at this once we add the blue light. And if you compare this to the dark control, it's quite similar and quite comparable. So we did this for 48 hours. We also did this experiment for 72 hours. We're sort of at the limits a little bit with these HeLa cell lines after 3 days, keeping them in the dark without any sort of good source of food and nourishment. But suffice to say, we could keep the signal off with green light. We could restore some of it, right, with blue light, and which was similar to the dark. And so that was very promising. And then we want to look at the effective reversibility of this construct. So like I said before, we could add green light, keep it inactive. Green blue, we could see gene silencing. But we wanted to see whether we could add green light and then not a few hours later, but 24 hours later add blue light. So you can think of the RNA being inactive for about a day, and then a day later we activate it, right? Like what's going to happen? Is it actually still going to work? Are we going to get some activity? And that's exactly what we see. We do see activity as shown in the purple here. It's not as pronounced as sort of these dark blue, but that's expected because, you know, these RNAs have been through sort of a more prolonged period of inactive form. And then we want to do this 1.5, so 1.5 green, blue, green, which is similar to that UV vis UV that I showed previously. So inactive, active, inactive. So we would expect some level of, or we would expect it to be inactive. And that's exactly what we see is highlighted in the light blue greens. And then furthermore, green, blue, looking at its ability to go through 2 rounds of cycling, and we expect to have some level of activity because we're finishing with a blue, and that's exactly what we see. And you can see that the levels are not as strong, of course, as some of the other ones, but that makes sense because it's gone through 2 states of inactive conformation with the green, and then our dark control, as I'm showing you here. So we're very pleased to see that we could reversibly control this siRNA. And we could observe very similar and same trends with the 72-hour experiment. So I won't kind of walk through every single detail, but the trends are the same. So this was really exciting. And then so for the last maybe 5 to 7, actually probably 7 to 8 minutes, I want to talk to you about some of the work that we've now transitioned to looking at whether we could do this in an in vivo system. And so this involved work with my collaborator, Professor Danina Simmons, and then our recent graduate, Mackenzie Mateus, a graduate student who spearheaded this project. Now, we wanted to look at Japanese Medaka, and we want to look at embryos. What are Japanese Medaka? They're small freshwater fish that have desirable features for use as a vertebrate model. So we have an aquatic lab here at Ontario Tech with my collaborator, Danina Simmons. But what's nice about these fish is that they spawn eggs daily. There's a number of useful strains. What you see here on the left is a transgenic enhanced GFP Japanese Medaka that we purchased from Japan. So you can see that they look, well, they look really pretty, but from an assay to be able to quantify fluorescence, this could be very handy. The genome is completely sequenced. The other important thing is that they have egg transparency, so that's really useful. So with our light experiments, we expect light to go inside of the embryo. And here's just a little brief video of one of the embryos. This is at day 8, and you can see the fish starting to develop. But the important thing I want to highlight is that the chorion is transparent throughout this. Throughout the 10-11 days. This is day 8, so this really gives us a good feel or good opportunity to add colored lights to these embryos in this in vivo system. Now, we synthesize, of course, our tetrafluorinated azobenzene siRNAs. So we have, in this case, we have wild type targeting the GFP. We have this F-siRNA, which now has azobenzene, the tetrafluorinated one, at the same location as the previous study. And then we developed a scrambled sequence here that has the same composition number of bases as these strands here, but in a really like a mixed jumbled up format, again, to just ensure that we're getting target specificity. And this is really hard to see here, but this is like a nano injector. And right here is the single cell egg at day 0. And again, it's really hard to see, but we're adding, or Mackenzie's adding 0.5 nanoliter siRNA to this egg. So it really requires incredible amount of fine tuning and control to deliver these eggs and puncture it just enough to not kill it and administer that siRNA. But let's look at some of the data. All right, so what I have here is our model again, active, inactive. And what we have here are some of the bioimages of the embryos, and this is after 24 hours. So, basically, let me walk you first through the legends. So we have something called azogreen. So this is where after injection, Mackenzie added green light to the embryos and then kept them in the dark. And then we read sort of the expression level of EGFP every 24 hours. So every 24 hours, because the cells were, or the embryos were subjected to some degree of like ambient light, et cetera, he would sort of administer a bit of green light to the cells just to ensure that they're in their inactive state. Azo green at blue at 72 hours. So what this means is that this was kept under green light, and then the blue light was not administered until at 72 hours. Azo blue at green at 72 hours is a reverse. So blue light was administered, and then at 72 hours, green light was administered. And then azo dark is just the active azobenzene compound in the dark phase. And then wild type siRNA, as I'm showing you here. So we can see here that the scramble, there's no effect happening here. And then what you can see here is that with wild type, you can start to see some gene silencing, and then maybe start to see a little bit with the azo dark as well. The azo blue to green is in the active state as well. And you start to see a little bit, but nothing really significant happening because it's only been 24 hours. And these all look pretty much the same because we're just at the start of the experiment. Now I'm changing to 48 hours. And what you can see now is a clearly defined reduction in wild type at 4 nanogram, 8 nanograms. And you start to see also downregulation of azo dark, okay? And you see downregulation also of azo blue at this concentration as well. But you can see that the azo green and the azo green at blue, these two areas here are not showing any reduction in EGFP, suggesting that they are inactive. So that's promising. That's good. And if you kind of eyeball up here, you can see here that our scramble is not doing anything. Of course, our controls, and you can see sort of good, strong fluorescence up here, and then you can certainly see reduction of fluorescence with these two that I just mentioned. Moving on to 72 hours, now you can start to see much more pronounced reduction of EGFP for the blue to green, and then for the blue to green. So remember, the blue to green, we haven't actually added the green light yet. Okay, just after this measurement, then we start adding it. So we expect this to be fairly comparable, and that's exactly what we see. And then more importantly, the azo green at blue, they're remaining inactive. So now right after this experiment, we're doing the reverse. We're now adding blue light to the embryos that were treated with green light, and then green light to the ones treated with blue light. So at the next time point, 96 hours, we might start to see a shift in activity, asking the question if it's reversible. So let's look at this now. Certainly we can still see pronounced gene silencing with the azo dark and wild type, more attenuated at 8 nanograms because it's more concentrated. But with the azo blue to green, in the previous experiment, I'll just flip back quickly. You can see we're seeing activity silencing, but now these bars are going up, meaning that they're starting to become inactive. And then I'll just flip back. You can see these bars here were inactive, but now they're starting to go down, meaning that we're starting to turn on the siRNA after 3 days of inactivity. And then we do this for 120 hours. Okay, again, silencing with azo dark and wild type. Here's our azo dark. You can see it's not as fluorescent as these other guys here, but you can see here that the blue to green is starting to go up, and then the one that was inactive is now coming down. And then the one that azo green really is quite inactive compared to the other guys. So this is really certainly exciting to see this reversibility trend working in these Japanese Medaka embryos. Now we've gone 120 hours, right? Which is I think about 5 days. So at some point, some things to keep in mind. The EGFP has a half-life of about 26 hours or so, and then these siRNAs, they have a certain half-life as well. We didn't quantify that, but at some point, this is not going to last forever. So 144 hours, we start to slowly see the levels rise, but it's really what we think is attributing to just sort of natural breakdown of the siRNA inside the cell. We didn't stabilize them with any stabilizing modifications. And if we keep going up to like 168 hours, we certainly see that these levels are almost identical. We certainly see these levels not as high because these were sort of always in the active stage. Going down 96, 92 hours, everything is sort of back up to normal, okay? But the trend that you can see is that we've injected and then we've gone down, 120 hours is sort of the sweet spot with silencing, and then the EGFP went back up. So this is sort of data that we're looking to publish really soon, but we're really excited that we can do this in an in vivo system. So just to summarize, so we've gone from this system here where we have azobenzene, we can inactivate it with UV light, visible light can reactivate it. Like I said before, UV light is not certainly ideal. So we moved to this tetrachlorinated group. Here's the little red box that Matthew developed, keeping like the eggs or the cells happy under red light. So we moved to this tetrachlorinated group. We redshifted it, which was great, but it had low thermal stability. So we had to keep that red light on. So then we moved to the tetrafluorinated group where we could use green light and keep it inactive for long periods of time. And then at any desired given time point, we could add the blue light to it. So future directions, now that we know this works in vivo, we want to try to understand better the mechanism of actions, the effect in vivo. We're really interested in other oligonucleotide designs, like not just siRNAs, but can we control other nucleic acid-based structures with this tetrafluorinated azobenzene group? And this certainly offers the potential to study these genes in vivo, like in real time, and study multiple genes simultaneously. So you can imagine if you're trying to understand a particular genetic or pathway, you could kind of turn on and off genes or at different potency levels to really try to understand their function in real time. So with that, I just want to spend just some time just talking about the people doing the work. So this is Matthew. So like I said, he really started this project as a graduate student, and then Mackenzie sort of took it over and looked at the in vivo effects with the Japanese Medaka. I'd like to just thank the funding agencies. I'd like to thank Aaron, and and if there are any students or any people are interested in potential grad studies or postdocs, I do have some openings. So if you're interested, do contact me after this webinar. I'd be delighted to chat with you. And with that, I'd be happy to take any questions. Thank you.
That was a great talk. I'll just remind everyone that you can use the Q&A function to ask some questions. So I just have a general kind of question while we're waiting to get some more in. I mean, I think that what you're doing is quite unique in terms of the context of the OTS and what people are doing with your kind of light up science. And I'm wondering, like, how did you get into that field, and where do you think that is going in terms of projects that you may have open for potential students in your lab?
Yeah. Okay. Yeah. Good. Yeah. Good question. I mean, I really, if you asked me, X number of years ago, like, would I be working with photoswitchable groups? And I mean, the answer would have been no, because I really didn't know anything about them, right? But how did I get into it? Well, it was really based upon that flexible linker study that we had, and then, then just me sort of understanding peripherally the power of azobenzene, and then taking some risks and just trying things that could have the potential for high impact and payoff, right? So, the azobenzene certainly was attractive for like peptides and a lot of other things, but its utility embedded within the RNA hadn't been explored. So, we just tried it, right? And it was really exploratory, I would say. And then things looked to work with our proof of concept. So we then sort of built upon that through different derivatives, right? So that's sort of the backstory.
Hmm. That's so neat.
Possible. I've also got a question. Yeah, it's a beautiful word. I actually had a question more on the molecular properties of of the azobenzenes along the siRNA. Uh, so because you're replacing two actual nucleobases with the azobenzene, do you see any significant changes in your TM and any like dependence on where that azobenzene on melting temperature of that duplex itself?
Yes, yes, yes. So that's a great question. So like looking at the biophysical properties of these azobenzene containing siRNA. So yes, we do see a drop in TM. Generally speaking, it can range from 10 to even 15 degrees, right? Because we're really replacing two nucleotides. And we've tried some experiments where we try to look for differences in TM in the cis or trans conformation, but we have trouble actually characterizing that because sort of as we heat up or melts, then it's hard to keep it in the cis form, right? But to answer your question, yes, we do see a drop of about 10 to 15 degrees, but it's okay because our target TM is around 70, 75 degrees. So we're going to around 60 degrees, maybe 65, which is not bad. So it still sort of functions. We're not sort of at that threshold where the duplex would sort of come apart at sort of biological temperatures.
Yeah, that's really cool. And if I could, I've got another question as well.
Mhm.
So you're moving more towards using different wavelengths of light, shifting it based on modifications on the azobenzene ring itself. So when it comes to using a less transparent animal, like say moving to mice models, like at the very least you need to go to near IR. Do you think that's something you'd be able to push the system towards, or
Mm.
would you implement another system for this?
Yeah, I mean that's a great question. Yeah, so we're not in the sort of the IR range, but I think that would sort of be kind of the next step and the next goal, maybe from a practical point of view, right? In terms of trying to use this sort of in a sort of a mammal model that doesn't have sort of a translucent body. Um, with respect to maybe applications like sort of down the road, and I guess we're here like with OTS, right? So, maybe this could be useful for sort of like eye therapy or through injection of administration of an oligo, and to kind of control the activity, maybe the patient would wear like different glasses at different times or something like this, right? So that could be useful as well if we can't move things to the near infrared or maybe skin topical treatments. And advances in medicine are also occurring where you can have like LED sort of lasers going into various tissues, although that is a little more intrusive. It's not like we wouldn't want to have a situation where you have to open up the patient and then add light, and then that's certainly not feasible, right? So to answer your question, yes, we would like to eventually go to the NIR. That would be great, but with what we have, we think we could make something useful out of it.
Like it's also be very interesting if you pair this with, for example, another system like magnetic nanoparticles for like that you can oscillate, have IR radiation in local parts in
Yeah,
vivo,
yeah.
but just very cool. We've also got a question in the chat now. Do you think the ASO modification will be compatible with other common chemical mods like LNAs? And like, do you think you maintain the photoswitchability with the increased affinity between strands when you have these modifications?
That's a great question, actually. Yeah, so we've only actually tested this modification with native RNA, right? And if we think of like the common modifications used in, let's say, siRNA development, a lot of the main players are like 2' fluoro, 2' methyl, and those certainly increase the TM as you mentioned. So, I think there needs to be, we have to be mindful of the TM and balance TM with azobenzene design. We haven't done a thorough analysis, but you're right. We might have too many stabilizing modifications, especially if we put things like walk nucleic acid, and then maybe that threshold is too high to allow for that switch to happen, right? But I do believe that sort of chemical modifications would work with the design placed at strategic locations, and that's something like we'd like to sort of explore in the future.
Totally. Like a lot of my work, a lot of the work my PI does is based on using specifically nanostructures with this. So like introducing azobenzenes, introducing this sort of forced change in morphology and implementing that on like larger structures is something also would be cool. So have you thought about using this in different ways, using mRNAs or using longer oligos than siRNAs themselves?
Yeah, yeah, absolutely. Yeah, we've done a lot of work on siRNAs, but yeah, we want to explore the effect of the scaffold on other oligonucleotide designs. So, I mean, for example, you're describing kind of very large, large sort of macromolecular complexes, but I think as a sort of what we'd like to do is maybe be able to control like even things like hairpin formation, hairpin function, right? Like we have a model system where we could control between like hairpin and sort of elongated forms. Or you could imagine if we have something like this, we could append this onto like a longer RNA, like an mRNA, and develop some kind of cap or some way to turn on and off the mRNA, right? So I think it's amenable for a variety of different oligonucleotide structures and not just sort of double-stranded RNAs. And it could be useful for maybe for even things like antisense or or trying to control like large complexes, like through these three-dimensional things like three-way junctions, for example. So I think there's a lot of room to explore in the oligo field with this. I would like to say though that one of the challenges, though, with this complex, I went through the synthesis, is that it's the yields are all really good, but it's sort of an 8-9 step process. So it takes a bit of time and expertise to make that, which is not, of course, which is doable, of course, but in terms of being able to scale that up and get enough material to make a wide variety of substrates still remains a little bit of a challenge. So we'd like to find ways to improve the synthesis or scale it up in the future as well, so we can answer those kinds of questions that you talk about.